Good morning. In this video, we will discuss about metabolism of chylomicrons and VLDL, very low density lipoprotein. So how these lipoproteins are synthesized, how they execute their functions, and after serving the functions, what is their fate. All those things will be discussed in this video. First, we will start with chylomicron metabolism. These chylomicrons, which are rich in triglycerides, they are synthesized mainly within the intestinal mucosa. It is the only lipoprotein which contains all the different types of apolipoproteins, ApoA, B48, ApoC and ApoE. It mainly helps in the transport of dietary lipids, that means mainly the dietary triglycerides from intestine to the peripheral tissues. Now we will see how they are synthesized in the intestinal mucosa and how they help in the dietary lipids, dietary triglycerides from intestine to peripheral tissues in detail. So to start with synthesis. The synthesis takes place within the intestinal mucosal cells. Inside the intestinal mucosal cells, first the apoproteins required for chylomicrons will be synthesized within the ribosomes which are attached to the rough endoplasmic reticulum. So what are the apoproteins present in chylomicrons? ApoA, B48, ApoC and ApoE. Okay, in that ApoA and B48 will be initially synthesized by the ribosomes within the intestinal mucosal cells. After their synthesis, both these apoproteins will be transferred to the smooth endoplasmic reticulum. Within this smooth endoplasmic reticulum, a protein called as microsomal triglyceride transfer protein, they will be transferring the lipids to these apoproteins and converts it into lipoprotein, which is nothing but chylomicrons. Now these chylomicrons, by a process called as exocytosis, it will be thrown out of the cell into the lymphatic drainage. Through this lymphatic drainage, it enters into the systemic circulation. This is how the chylomicrons are synthesized in the intestinal mucosal cells. Okay, so chylomicrons, any lipoprotein, it is made up of both lipids and proteins. Initially, the proteins ApoA and ApoB48 will be synthesized in the ribosomes, which are attached to rough endoplasmic reticulum. Then they will be transferred to smooth endoplasmic reticulum. In the smooth endoplasmic reticulum, by the action of this MTP protein, lipids will be loaded and it will be converted to chylomicrons, and that chylomicrons will be thrown into the lymphatic drainage through which it reaches the circulation. So the newly synthesized chylomicron which reaches the circulation, they will be having only ApoA and ApoB48. So this one is called as nascent chylomicron or immature chylomicrons. They will become mature only after receiving the remaining apolipoproteins, that means ApoC and ApoE. So this ApoC and ApoE, they usually get it from HDL in the circulation. After receiving this ApoC and ApoE, the nascent chylomicrons becomes mature chylomicrons. Once it becomes mature, they go in contact with the peripheral tissues. When it comes in contact with the peripheral tissues, ApoC2, which is present on the surface of chylomicrons, will activate lipoprotein lipase enzyme, which are present on the capillaries on the surface of the peripheral tissues. So once this LPL gets activated, as the name says, lipoprotein lipase, they will be hydrolyzing the triglycerides present in the lipoprotein chylomicron, and they will be taking up all the fatty acids. Okay, so after serving these peripheral tissues like skeletal muscle, heart, mammary glands, etc., then they will be moving to the adipose tissue, where there also by the same mechanism, ApoC2 activation of LPL, and this LPL will be deriving all the triglycerides from chylomicrons to the adipose tissue. After donating all the triglycerides from the chylomicrons, the function of ApoC2 will be over. So this ApoC2 along with ApoA will be transferred back to HDL molecule. Okay, so now the chylomicrons, after losing its triglycerides and after losing its apoproteins like A and C, it will be transformed into chylomicron remnant. Okay, so this remnant, what they will be having means, inside the chylomicron remnant, we will be having the dietary cholesterol molecules and fat soluble vitamins, whereas on the outer surface, we will be having ApoB48 and ApoE protein. Now, with the help of this ApoE protein, This chylomicron remnants will be taken up by the liver. Okay, so in liver we have specific receptors called as LDL receptor or LDL receptor related protein. So these two proteins will identify the ApoE, they will bind to it and they will engulf the chylomicron remnants, and it will be completely catabolized within the liver with the help of an enzyme called as hepatic lipase. So this is how chylomicrons are metabolized in our body. Okay, so to sum up, the chylomicrons are synthesized in the intestinal mucosal cells. Okay, so after synthesis, they will help in transferring the dietary lipids from intestine to the peripheral tissues like skeletal muscle, heart, and finally to the adipose tissue. After donating all the triglycerides to the peripheral tissues, the chylomicrons will shrink in size to form chylomicron remnants. That remnants will ultimately be destroyed by the catabolized by the liver. Okay, so that's it about chylomicron metabolism. Next, we will see about VLDL metabolism. So this VLDL, which is also rich in triglyceride, is synthesized within the liver. Okay, so this VLDL is made up of all the apoproteins except ApoE. That means it has ApoB100, ApoC and ApoE. It helps in carrying the endogenous lipids, mainly triglycerides and also cholesterol, from liver to the peripheral tissues. Okay, so this is a brief introduction about VLDL. Now we will see in detail how this VLDL molecule is formed and how they help in carrying this endogenous lipids in detail. So first, synthesis. The metabolism of VLDL is almost similar to chylomicrons except for few differences. So what are those differences? Means the first difference is the chylomicrons are synthesized in intestinal mucosal cells, whereas the VLDL will be synthesized within the hepatocytes. That is the first difference. Okay, so within the liver, here also first the apoproteins required for VLDL will be synthesized. That means mainly here ApoB100 will be synthesized in the ribosomes which are attached to the rough endoplasmic reticulum. Now this ApoB100 from rough endoplasmic reticulum, it will be transferred to smooth endoplasmic reticulum. So inside the smooth endoplasmic reticulum, a protein called as microsomal triglyceride transfer protein, they will be transferring the endogenously synthesized triglyceride into this ApoB100, so that converting it into a lipoprotein called as VLDL2. Now this VLDL2 from smooth endoplasmic reticulum, it will be going to the Golgi apparatus, where it will fuse with triglyceride rich droplets and it gets converted into VLDL1. Now this VLDL1, by a process called as exocytosis, it will be thrown out directly into the systemic circulation. Okay, so this is how VLDL molecule is synthesized and excreted from the hepatocytes. This synthesis and excretion of VLDL from liver cells can be inhibited by all these factors. Okay, so thus all these factors may have a preventive role in hypertriglyceridemia or a causative role in fatty liver. So they prevent the production of VLDL from production or excretion of VLDL from the liver cells. Now we will come back to the metabolism. Okay, so after it reaches the systemic circulation, how it serves its function and how it is catabolized, we will see in detail. Okay, so this metabolism is again similar to chylomicrons only. So the newly synthesized lipoprotein from liver, we call it as nascent VLDL molecule. So this nascent VLDL molecule will be having only apoprotein B100. Okay, so they will become mature only after receiving ApoC and ApoE from HDL molecule. So when it receives ApoC and ApoE from HDL, it becomes mature. Once it becomes mature, they go in contact with the peripheral tissues. When it comes in contact with peripheral tissue, ApoC2 present on the surface of VLDL molecule will activate lipoprotein lipase, which is present on the surface of peripheral tissues like skeletal muscle, heart tissue, mammary glands, etc. So this ApoC2 activates LPL. That LPL in turn will hydrolyze the triglycerides present in that VLDL, and it will be taking up all the contents of VLDL. So after donating to the peripheral tissues, the VLDL molecule will move to the adipose tissue and further dump all its triglycerides into the adipose tissue through the same mechanism. Okay, so once it delivers most of its triglyceride contents, the VLDL molecule, once the triglycerides are transferred from VLDL to the peripheral tissues, the function of ApoC2 will be over. So this ApoC2 gets back to the HDL molecule. So after losing the triglyceride content and also the ApoC, the VLDL molecule will shrink in size and it will be converted to its remnant, which is otherwise called as intermediate density lipoprotein, which is shortly designated as IDL. Okay, so this is up to this. It is almost same like chylomicron metabolism. Okay, so nascent will be formed. They will be receiving the apoproteins, remaining apoproteins from HDL. They will become mature. Then they will be donating the triglyceride to the peripheral tissues. After donating, they will be converted into remnants. Okay, so now this VLDL remnant or IDL, which is formed during VLDL metabolism, will have two different fates. One is like chylomicron remnant. With the help of ApoE, this will also be taken up by the liver cells and it will be completely catabolized. Or alternatively, what can happen means this IDL, it can further lose its triglyceride molecule, it can further lose its apoprotein E and can be converted into LDL molecule, ApoB100 cholesterol rich LDL molecule. Okay, so these are the two fates of VLDL remnant. Okay, so one like chylomicron remnant, with the help of ApoE, it can be taken up by the liver cells and it can be catabolized, or it can further lose its triglycerides, ApoE, and can be converted into cholesterol rich ApoB100 containing LDL molecule. Okay, so that's it about the VLDL metabolism. So it is synthesized in the liver. From liver, the endogenously synthesized triglycerides will be transferred to the peripheral tissues. After transferring it, it gets transformed into intermediate density lipoprotein, and this intermediate density lipoprotein will either be catabolized in the liver or it will be converted into the LDL in the circulation only. Okay, so that's it about chylomicrons and VLDL metabolism. Thank you.